Alright guys, we are going to review Unit 4. This is going to be over the genetics, so it's the genetics part 1, genetics part 2, and the genetic disorders. Okay, so we're going to take a look at this, and this is your last unit review test before you take your lab final and your lecture final. So let's get into genetics. Now, first of all guys, this brings up an argument when we look at genetics of nature versus nurture. Now remember that genetics is actually the study of heredity. It's what's in your genes, and it's what gets passed on from one generation to the next. However, the nurture side looks at environmental factors. We are actually a combination of both. There are certain environmental factors that actually affect our genetics. Some of these include in some animals like temperature, diet, drugs that might be taken, because they can actually alter how genes are going to be expressed. So we are actually going to be a combination of both. Well, this brings us to Mendel. Mendel, guys, is the father of genetics. He's the one that studied pea plants. He was an Austrian monk, and so he was going through and looking at how genes got passed on from one generation to the next. So he came up with 3 laws. So let's talk about these 3 laws really quick, because you're going to need to be able to recognize them. The first law is the law of dominance. This says that there's going to be 2 factors for each trait. However, one is going to be dominant over the other, which we call the recessive. If the dominant trait is present, it actually masks or hides the recessive trait. The recessive trait's still there, you're just not seeing it. It's being hidden. It's masked. Okay, so this is what we call the law of dominance. The second law that he talked about is the law of segregation. Now, segregation means that we're going to separate. Now, remember that all organisms are considered diploid organisms. So organisms have 2 copies of a trait. That's what makes us diploid. So when this happens during meiosis, we talked about how those particular traits like separate during anaphase. We want to separate them, and that's what this law of segregation says. When we separate in the form of meiosis, we see that we're going to form egg and sperm. Egg and sperm are the haploid versions, meaning they only have 1 copy of the trait. The way to get back to the diploid is through fertilization. When an egg and sperm fuse together, we now return back to a diploid because you got 1 copy from the egg, 1 copy from the sperm. So this is the law of segregation. Now, the last law deals with the law of independent assortment. This one says that each pair of alleles is going to assort independently from all the other pairs. So when we look at this, chromosome 1 doesn't care what chromosome 5 is doing. Chromosome 5 doesn't care what chromosome 15 is doing. They're going to all line up independently. Okay, and so when we look at this, this allows for it to be random, and it gives us more variability in an organism. So now let's take a step further and let's look at Mendel's experiment and what he kind of came up with his crosses that he did. So when we take a look at this, guys, Mendel went through and he talked about his generations. He had a P generation, an F1 generation, and an F2 generation. So if we take a look at this, if we're talking about flowers here, we have big P is for purple flower, and we have little p for the white flower, which tells you that the purple is dominant to the recessive. Remember, in dominant traits, we represent them with a capital letter. Recessive, we represent with a lowercase letter. So when we look at this, the parent generation, the P generation, are true breeding parents, meaning that they're big P, and little p. They're purebred, which we call homozygous. Homozygous means you get the same thing. So homozygous dominant is the big P. Homozygous recessive is the little p. Now when you cross these in a Punnett square, you're going to see that all of the offspring in the F1 generation are what we call hybrids. They're a combination. They're a big P and a little p. However, they're still purple in color because purple is dominant. So when we see this, they are considered hybrids or heterozygous. Heterozygous means that we have 1 big letter and 1 little letter. Now, if we take 2 of these heterozygous individuals and we cross them, we're going to see that we're going to get mostly purple individuals. However, we will also see that you're going to get some of the recessive. So in the parent generation, if you notice here, that's the parent line, true breeding. We had a big P crossed with the little p. This resulted in the F1 generation, which caused hybrids, big P, little p. Now, when we take these hybrids and we cross them, we're going to see we get 3 dominant with the purple, and we get 1 recessive. However, guys, one of those dominants is homozygous dominant, big P. The other 2 are heterozygous, big P, little p, and of course the 1 recessive are 2 little p's. So if you need to review that, put the Punnett square together and see how this works. But guys, it is important to know what Mendel's parent generation is, his F1 and his F2, and you can find that here on this slide. So now we want to switch gears and we want to talk a little bit about some definitions or terms. So you're going to notice here that there's a lot of this versus this because we have some opposites that are here. Now some of this is a review because we have talked about haploid and diploid before. So let's look at diploid versus haploid. So diploid is where you have two full sets of chromosomes. We represent this with 2N. You are considered a diploid organism. You got half your chromosomes from your mom, half the chromosomes from your dad to give you a full set. On the other hand, haploid means they have half the chromosomes. Okay, this is represented by the letter N. Haploid for us would be like 23. You have 23 chromosomes in your egg and your sperm. The reason we have those haploid cells is through the process of meiosis. Now, this brings us to some terms like gene, allele, and gene locus. So guys, when we look at the gene is our unit of heredity. This is where we find the code for whatever the trait is. We also see that there are what we call alleles. Alleles are alternative forms. So you can have a dominant allele represented by a capital letter, or you can have a recessive allele represented by a lowercase letter. So this is why we get those different letters. Also, you have the gene locus. The gene locus is the location of the gene. So we have gene, which is the hereditary unit. Allele are different variations of that gene, and then the gene locus is its location. Now this brings us to what we call autosomes versus sex chromosomes. Now I have a little diagram here that's from your notes that kind of helps with this. When we take a look at this, autosomes are your first 22 pairs of chromosomes. Okay, so when we look at this in your skin cells or your body cells, you have technically 44 chromosomes. That's 22 pairs of autosomes. Okay, in a gamete, whether it's egg or sperm, you only have 22 because we have that number. There's no pairs here. Your sex chromosomes is the 23rd pair. You only have 2. If you're female, you have XX, and if you're male, you have XY. So the sex chromosomes, guys, determine your gender. So you have, again, 1 pair for a total of 2. However, in your sex cells, the egg and sperm, you only have 1. It's going to either be 1X or 1Y. This allows for when the two fuse together, an egg and sperm, then that's when you get your pairs back again. Okay, so this little chart just kind of gives you an idea. Pay attention to the 44 or 22 pairs, or just 22 total when we talk about autosomes. Sex chromosomes, you have 2 or 1 pair, and then you only have 1 in your gamete of sex chromosomes. All right, dominant versus recessive, we've already hit on this, guys. Dominant, of course, is represented by the capital letter. It's the allele that's going to be expressed in the heterozygote stage. So if it's like big T, little t, you're going to see the big T. It's what is expressed. On the other hand, with recessive, recessive is represented by the lowercase letter, and it's hidden or masked in the heterozygous. Okay, when you have big T, little t, you don't see the little t. It's hidden. The only time you see the recessive trait is if there are 2 little letters, little t, little t. Well, this brings us to heterozygous and homozygous. I've already used these terms in this kind of presentation, but we'll review them again here. Homozygous means that it's a purebred, the genotype that contains 2 identical traits, whether it's big T, or little t, little t. Heterozygous, on the other hand, is the hybrid. It's the combination. It's going to have 2 different letters, so big T, little t. Also remember that heterozygotes are going to be seen in that F1 generation that Mendel tested, which you can see on the previous slide. So the last group of terms we want to talk about is phenotype versus genotype. Guys, remember the genotype is the genetic makeup of the individual, whereas the phenotype is the physical appearance. The genotype is represented by letters like big T, little t. However, the phenotype would be that you're telling me that the individual is tall or short. So let's do some application of this law of segregation that we talked about earlier. We want to kind of set up Punnett squares. This is the whole point of this. So if the parent genotype is big T, little t, this means they are heterozygous. Then we have 2 possible gametes. When we separate these, you have a big T that's separated from the little t. So let's look at a monohybrid cross where we're looking at 1 trait. So I'm going to cross 2 parents that are heterozygous, big T, little t, and big T, little t. When I set up the Punnett square, we're going to separate these. The first big T is going to go on the first column. The first little t is going to go in the second column. The second big T is going to go in the first row. The second little t is going to go in the second row. So this creates our Punnett square. So now what do we put in the boxes? Now remember, you're going to drag what's at the top down and what's at the side over. So in this first box, you're going to have big T, big T. The next box, you're going to have big T, little t. The following one, big T, little t. And the last one, little t, little t. Now remember, with these, you are supposed to actually be putting the capital letter first. So now this brings us to the probability. Now what can we do with this information? Well, we're going to look at 2 things where we can look at the sum rule of probability and the product rule. In the sum rule, you're going to do like an addition. Okay, you're going to add it together. On the other hand, for the product rule, we're going to do some multiplication. So let's look at the sum rule first. This would be, what do we get with this or this? So when you look at this with the or term here, you're going to do addition. So what are the chances that you're going to get a short individual? Well, it's one box. This one box is 1 out of 4, so this is 25%. However, what are the chances that your first plant is short and your second plant is short? This is where the and comes in, where I'm actually going to do the multiplication. So this would be 1/4 times 1/4, which equals 1/16. So pay attention versus the addition rule versus the product rule, the sum versus the product. Now, this is great doing Punnett squares when you know what both parents are, but what if you don't know what one parent is? This is going to bring us to what we call our test cross. The purpose of the test cross is to determine the genotype of an individual expressing the dominant trait. Because guys, if they have the dominant trait, this means that they could be big T, homozygous dominant, or they could be big T, little t, which is heterozygous. That's going to be expressed the same way as being tall in this case. So we don't know what they are. So we're going to do 2 Punnett squares to see what our results are. So we can use the big T times a homozygous recessive, or we can do the big T little t times the homozygous recessive. This is going to give us different Punnett squares. So in the first case here, you have 100% of the plants being tall because they're all heterozygous. But in the second set, you only have 50% being tall and 50% being short. So what you need to do is you got to look at the offspring. If no offspring that were produced in your experiment are short, then the individual was big T, big T. However, if you get both tall and short individuals, then then the individual was big T, little t. So this is just reviewing the monohybrid cross and the purpose of a test cross. So let's take a look at predicted results, because you're going to need to be able to predict these using the Punnett square. So if we're looking at the first set here, if the parents were homozygous dominant crossed with homozygous recessive, what are the chances of the offspring's phenotype? Well, they would all express the dominant trait. In this case, if we're looking at T's, that would be considered tall. Okay, so they would all be big T, little t, all tall. However, if you take 2 parents that are heterozygous, big T, little t, you're going to get that ratio that's a 3-to-1. 3 dominant to 1 recessive. Now, what if you take a big T, little t times 2 little t's? This is where you're going to get your 50-50. This is a 1-to-1 ratio, so you'd have 1 dominant to 1 recessive. Okay, so these are what we would look at for monohybrids. Now, dihybrids are a little more complicated. With dihybrids, we're looking at the 2 traits. So with these 2 traits, if I was to take a completely homozygous dominant crossed with a completely homozygous recessive, we're going to get the same results. All are going to express the dominant trait. So they would all be tall and they'd all be yellow if this was the case. All right, so when we're looking at this, you're going to see that they're all going to be heterozygous for the traits. But now if I take 2 heterozygous individuals, we're going to get this weird ratio. We're going to get 9 that do both dominant. We're going to get 3 that is one of the dominants, like tall, but they're going to have the recessive for the color, which would be green. They'll have 3 for short with the dominant yellow. They'll have 1 that's completely recessive being short and green in this case. Okay, so this is what we're looking at for that cross. Now, what if I take a heterozygous cross with a homozygous recessive? This is where we're going to actually get the 1-to-1 to 1-to-1 ratio, exactly with what you saw before. All right, so when we're looking at this, this is to give you an idea of what you're seeing without actually having to do the Punnett square. But during your exam, if you need to set up the Punnett square and do it to get the answer, go ahead and do that. You can get scrap paper and do this. However, this slide is just going to help you see what the ratios should be right off the bat. So now what we want to talk about is we want to talk about some things that kind of go against just our normal Mendelian genetics. This is where we get a little more complicated. So the first thing I want to talk to you about is incomplete dominance. When we're looking at incomplete dominance, this is where one allele is not completely hidden by the other. So the best example of this is with the flower colors. When you cross a red flower with the white flower, you get the pink. It's not completely dominant over the white, so it's a mixture. Okay, so we see both. However, if you take 2 pink flowers and you cross them, you're going to get 1 red, 2 pink, and 1 white. This gives you a 1-to-1 ratio for incomplete dominance. So the best example here is the flower color. The heterozygous big R little r is pink. So now let's switch gears and look at codominance. Codominance, one of the best examples, is where both alleles are being expressed equally, and AB blood type is one of our best examples. So when we take a look at this and we do a Punnett square with it, if I take an individual who has AB blood and I cross it with somebody who has AO blood, you're going to get where you have some A blood, you have some AB blood, and you have some B blood. You don't have any O because O is the one that's completely recessive. If I take an AO times a BO, you're going to get all 4 blood types. You're going to get AB, A, B, and O. So these Punnett squares are a little different because A and B are expressed equally. They are codominant. The next thing we want to look at is an X-linked disorder. X-linked disorders are located on the sex chromosome, specifically the X chromosome. This is going to affect males more than females because females have 2 X's, whereas a male only has 1 with that Y. A great example of this is red-green color blindness. When you take a look here with red-green color blindness, you can see that the mom is the XX on the top. She's a carrier of color blindness. She's big B, little b. The dad is XY on the side, and he is not. He's got normal vision. So you'll notice that the X, a big B, and the Y, he's got normal color vision. So when we do this with an X-linked cross, you have to make sure you put your X's and Y's in there versus boys versus girls, because that can make a difference on what the question's being asked. So in the top boxes, you have girls. One has complete normal vision. The other also has normal vision, but she's a carrier for color blindness. For the boys, you get one boy over here who has normal vision, but then you have one boy that is colorblind. Now you can see though how the boys are affected more by the colorblind trait. So this is an X-linked trait. The next we want to look at is paleotrophy. Paleotrophy is where one gene affects many traits. So one gene can alter a lot of different things that we see in the body. And one of the best examples of this is in your notes with Marfan syndrome. Another was also sickle cell anemia. Because with Marfan syndrome, the defective gene is the protein for collagen, and collagen causes a lot of things to go wrong if it's not working properly. It can cause abnormal connective tissue. It can cause you to be tall and thin, have long arms and legs, being nearsighted. Again, none of those are life-threatening. However, the last 2, weakened aortic vessels and respiratory issues, can be life-threatening, and people could die with Marfan syndrome here. So we got to see that it's going to be one defective gene, but it's affecting multiple traits. Now the opposite of this is polygenic inheritance. Polygenic inheritance says there's many genes only affecting one trait. The best examples here are skin pigmentation, like skin color, and height. So here's an example here with the skin pigment. If you have all dominant traits, you have all 6, because it's 3 different genes. 3 times 2 is 6. If you have all 6 dominant alleles, you have very dark skin. On the other hand, if you have all 6 of recessive alleles, you have very light skin. If you're a combination of both, you have medium skin. So this is what gives us that bell curve that allows us to see all the different variations. Alright, so let's look real quick at the gene mutations. With gene mutations, there can be a change in the chromosome number, which you can see here. Now, this picture right here is a karyotype. Karyotypes can be used to diagnose certain disorders like an extra chromosome, or maybe missing a chromosome like a trisomy or a monosomy. It can also be used to look at if the chromosomes look different, like you can see here. The chromosome structure is different because 5 is missing a piece. With a deletion or something like that, this would be something like Williams syndrome or Crowdy-Chat, which we talked about in class. Now, going back to the idea of an extra or missing chromosome, this happens due to non-disjunction. So if you notice over here, you can see that they do not break apart, and it's labeled in the diagram here with non-disjunction. Because those chromosomes are not splitting apart, then the whole thing is going into one cell, which gets the extra, but then this cell over here is missing one. So over here we have an extra copy, and this cell has one less. This is non-disjunction. Now this can occur in meiosis 1 or meiosis 2. That's important to note. Alright, we also see that we can see a change in the chromosome structure. This is going to give us different terms that we need to be able to identify based on the definition. If a piece of the chromosome rejoins in a reversed or opposite direction, that's called inversion. Okay, and you can see that here, inversion. If a piece of the chromosome rejoins to a totally different chromosome, to a different chromosome altogether, that's called translocation. It moves to a new location. If there's a loss of a chromosome piece, that's a deletion. And then, of course, if the segment of a chromosome gets repeated, that's a duplication. So be able to identify and match these up, because this will be a type of matching on your exam. All right, now this is going to bring us to the disorders. So I want to talk to you about some of the different disorders that we discussed in class and give you some kind of basic information about them. All right, so if we take a look here, guys, there can be abnormal chromosome numbers and autosomes. You can see in this first column here. Down syndrome, guys, is a trisomy 21. You need to make sure that you know that. It's 3 copies of 21. Also remember there's certain characteristics with Down syndrome. Of course, mental retardation kind of comes with that, but there's different degrees on that spectrum. They also have that moon-shaped face. You also see that they are going to have heart problems and also be more prone to leukemia. Just because of that genetic disorder. Patatus and Edwards are also trisomies. Patatu is a trisomy 13 and Edwards is a trisomy 18. Now, in the sex chromosomes, we can see that there can be some trisomies. The trisomies first with Klinefelters. Guys, Klinefelters is going to be XXY, which means that they are male. These males will have underdeveloped testes, meaning also there'll be a little bit of breast development due to the extra estrogen. And we see that they have pretty much normal intelligence, but it's going to be with kind of like learning disabilities, like dyslexia, things like that. They just might be a little slower at getting things, but there's no real true mental retardation that comes along with this. We also see that you can have a female who is XXX, that's a poly X, and Jacobs, which is XYY. Those are all 3 sex chromosome problems where they have a trisomy. On the other hand, the only monosomy that survives, mono means 1, is Turner syndrome. Turner means they have an X, but they're missing the other part. Now, it's represented here with like a 0. They have the X, but then they don't have the other chromosome. With Turner syndrome, guys, Turner syndrome is going to cause a woman to have shorter stature. She's going to have issues with puberty, menstruation, infertility, but pretty much pretty normal life besides those potential issues. They may also have some issues with heart problems, diabetes, things like that, but these are things that can ultimately be treated. All right, so now we're going to switch gears and we're going to look at traits that are due to being either recessive or dominant, whether they're autosomal or X-linked. So let's look at autosomal recessive first. Autosomal means that it's going to affect males and females equally. Recessive means you have to have 2 of the little letters in order to have the particular trait. Now guys, it's super important to know which disorders are autosomal recessive, which ones are autosomal dominant, and which ones are X-linked recessive. There will be a matching on knowing which ones they are. So autosomal recessive is little a, little a. This is going to be like Tay-Sachs. Remember, with Tay-Sachs, this is a disease that's going to be very prevalent in the Jewish community or Jewish people. It is inherited as an autosomal recessive disorder, and it causes the lysosomes not to function properly. We also see cystic fibrosis as autosomal recessive. With cystic fibrosis, this causes an abnormal thick mucus, and it's going to cause issues with their breathing and digestion. It's one of the most lethal genetic disorders for the Caucasian community. Phenylketonuria or PKU, this is going to be also autosomal recessive. You don't have the enzyme to actually be able to process a certain amino acid. This one is treatable. If you get tested and you see that they have this disorder of PKU, you can just alter the child's diet. The next column, though, guys, looks at the autosomal dominant. If you have this disorder, you can have big A, or you can have big A, little a. This is a higher chance to affect you. Again, though, with it being autosomal, it means it's going to affect male and females equally. So neurofibromatosis, this is going to be autosomal dominant. This is the one where you actually create tumors on the nerves of the body. It can also cause skin issues and abnormal bone growth. We also see Huntington's. Huntington's is going to be a neurological degeneration that takes place. So this one occurs later in life, and it happens when the individual is in their 30s or 40s, and it's going to start causing issues with their brain function. They start to lose the ability to do certain things, to talk, walk. They kind of go back into that infant stage. The last one here that's autosomal dominant is achondroplasia. This is a form of dwarfism. This is where the individual has a normal sized torso, but their arms and legs are short. There's also going to be skeletal issues that come into play with somebody with achondroplasia. The last group over here is the X-linked recessive. X-linked traits are going to affect males more often because females have that extra copy. So if a female gets this, she has to be X little b, X little b. However, with males, it would just be X little b, Y. Duchenne's muscular dystrophy is an example of this. We talked about how the muscles actually start to deteriorate due to the protein dystrophin not being produced, so it causes weakness. The muscles, however, look the same size because it gets replaced with fat. This becomes an issue if it affects the diaphragm muscle, which helps you breathe. We also have hemophilia on this list. Hemophilia is the inability to produce normal protein clotting factors in the blood, so they don't clot properly due to injuries. They could bleed to death from something pretty minor, like certain kinds of bruising and things like that. We also see color blindness. Color blindness is not a life-threatening disorder, but it is still X-linked. And the most common here is red-green color blindness. All right, so talking about these disorders, whether they're autosomal, if they're recessive or dominant, this brings us to that idea of pedigrees. Now guys, on these pedigrees, it's super important to kind of look at these and be able to tell if they're autosomal, to be able to tell if they're recessive or dominant. And here are some little tricks that kind of help with this. Autosomal recessive disorders, most affected children have normal parents, so they don't really have an issue in the parent side, but the kids have it. If they're heterozygous big A, little a, we don't see them having the problem. They're just carriers of it. Both male and females are going to be affected equally. So if we take a look at this, you can see that there's 2 females affected because females are represented by a circle. Males are represented by the squares. If they're colored in, like we see here in the blue, those are the ones that are affected. Also, if you'll notice, there's a line between individuals. That's a mating partnership, and they have kids that come out of that. So if we look here, this individual has a parent up here with little a, the other parent has a big A, and so none of the kids are affected. We then see another generation where nothing is affected there, but then you see in the last generation we see some more issues, and this is due to 2 heterozygous normal parents, big A, little a, coming together and producing 2 offspring that have the disorder. So this is autosomal recessive. Now, autosomal dominant looks a little bit different. You'll notice that there are more circles and squares filled in. Okay. Also, affected children usually have an affected parent because they have that capital letter. The heterozygous big A, little a is also affected here. So big A, and big A, little a have the disorder. 2 affected parents can produce an unaffected child, though, because they still could maybe be carriers of the recessive trait. Again, this affects males and females equally. The last one is X-linked recessive. If we look at the X-linked recessive, more males are going to be affected than females, and this is what you can see here with 3 of the boxes, the squares being filled in, which are males. An affected son can have parents who have normal phenotypes. Their parents could be normal because it gets passed down a lot of times from the mother to the son. For a female to have the disorder, she has to have both X's affected, so this is why it is a little more uncommon for females to have a disorder. So what we want to do here is we want to kind of quiz you on how to do this. So I'm going to go to some next slides and I'm going to ask some questions, and I know you can't answer me back, but I'll give you some time to take a look at them as we go through this video. So in this particular case, is this an autosomal dominant, autosomal recessive, or sex-linked recessive disorder? However, is it dominant or recessive? There are quite a few boxes and circles filled in. This is a dominant disorder. Now, one way to really take a look at this and see is look at this right here. I have 2 parents who are affected, which means that they have kids that are affected, but they also have a kid who is normal. If these were little a, if 5 and 6 were little a, then all they could have were kids affected. Number 9 is not affected down here at the bottom, so this tells you that it's a dominant disorder. So let's come up and find out what their genotypes are for these individuals. So if we look at generation 1, individual 2, this is a female, but she's normal, so she's going to be little a, little a. What about generation 2, individuals 5 and 6? They have the same type of genotype, so with these they're going to be big A, little a. And the reason we know they're big A, little a is if you take this Punnett square, they have one kid who's completely normal where the little a comes from, and they have 2 kids who are affected, so we can utilize the Punnett square. Now what about generation 3, individuals 8 and 10? Again, in 8 and 10, they are similar, but we only know that they have a big A. We don't know if they're big A, little a, because we don't have their kids. So we can't see what their offspring would look like. So we guys see how this works. This is what you did in lab 2 with your pedigrees. So let's go to the next one. So this one is autosomal, and the reason is if you look here, you have a parent you have two parents who are unaffected, but the girl has the disorder. So in order for the girl to have a sex-linked disorder, you're going to have to see that the dad was affected, because she has to get two X's that are messed up, which means she has to get a messed up X from mom and a messed up X from dad. Also, when you look at these, you have two parents who are not affected who have an affected child. This tells you also that it's recessive. So it's important to take a look at different parts of the pedigree. Okay, so now, saying it's autosomal and it's recessive, this is going to give us the key that little a is the afflicted. They're the ones that are colored in. Big A, and big A, little a are not affected. So individual or generation 1, individual 1 and 4, these are going to have little a, little a. Generation 2, individuals 3 and 4, they're normal, but they have to have big A, little a because they had children that have little a, little a. Generation 3, individuals 1 and 2, these are going to be individuals with little a, little a. So hopefully this is helping you kind of understand and see what's going on. So let's take a look at the last one. On the last one here, you can see that there's a major difference that's happening here. There's only squares filled in. Since there's only squares filled in, this is going to be an X-linked trait. Now, sex-linked, X-linked trait are all recessive. Okay, that's just how they are. So some justification of being X-linked and being recessive, as you can see here. Two normal parents creating a abnormal son. This tells you that the mom was the carrier. So let's look at generation 1, individual 1. It's right here. Up here at the top, remember X little a is going to be afflicted, X big A is not afflicted. So this is a male, which means they're going to have an X and a Y, and he has the disorder, so it's X little a Y. Now let's look at generation 2, individual 2. This is a female, so she's going to have two X's. However, she is normal, so she's going to have at least X big A. However, she has sons who are affected, so she's X little a. Generation 3, individual 3 is located here. It's a male and he's normal, so he's got X big A, Y. So let's take a look and talk a little bit about sickle cell and Fragile X disorder, because these were some things I wasn't able to cover in class. Sickle cell is important because this is a disease that is due to the production of a defective hemoglobin gene. So again, this is going to be one gene that's faulty that can cause a lot of problems, like we saw with Marfan syndrome. So if you carry the trait, you actually have a normal trait, which is the A, and you carry the S. This is the sickle cell gene. So you're heterozygous, which leads to normal life, but you have some of your red blood cells that are sickle shaped, like you see here in the picture. So it actually protects you from getting malaria. However, getting the disease is with two little s's. This is homozygous recessive, two faulty genes. So some of the symptoms with sickle cell is anemia. Of course, that's why it's called sickle cell anemia. It causes pain whenever your body goes into crisis because it's not getting oxygen to the parts of your body that need it. It causes joint bleeds and also organ failure, which can be life-threatening. The prognosis is typically a shorter lifespan, but again, it depends on the severity and it depends on where you live. In a developed country like the United States, you can receive treatment, but if you get this in Africa, you're going to have a harder time receiving treatment. Treatment is crisis prevention, preventing the body from going into a crisis state, and it's also just supportive. So this is the Punnett square that we would look at for sickle cell. We use different letters because it's kind of like a codominant structure, like we see with A, B blood. So if you notice here, the individual that has the big A, they're completely normal. They don't even carry the sickle cell trait. The two individuals that are big A, big S, they carry the sickle cell trait, but if they have two little S, or if they have the two S's, they actually have the sickle cell disease. So this is just giving you an idea of what you're seeing. The last thing here is the Fragile X. With Fragile X, guys, this is a disorder or syndrome that's due to the chromosome having an area that's ready to break off. And you can actually see it here and also in the karyotype. Very end of the X is fragile and barely hanging on, which means that it's going to have a harder time being expressed. So individuals with Fragile X, it's the second most common cause of genetic mental retardation after Down syndrome. Okay, so it's going to cause mental retardation. Males are institutionalized for mental retardation, outnumber females about 5 to 4. So it's actually really close for males and females with this because it still deals with that X and that X being fragile. Okay, so this is what we're looking at for Fragile X syndrome. Now, I wanted to go over those slides because those were at the end of the last PowerPoint and we just didn't get to them. Fragile X is not really on your exam, but I wanted you to know what it is. But sickle cell is on your exam, so take a look at that. If you have any questions or concerns about this exam, please feel free to come by and see me, email me, things like that. But you can rewatch this video over and over again. Also, the Quizlet cards will be posted as well with this video link. So please have a happy Thanksgiving, but also utilize your time to study a little bit for exams, since it's right after Thanksgiving that you'll be taking this in lab. Have a good day.